Well, thank you kindly for that introduction. I hope I live up to it. I too will be at the workshop tomorrow morning, and I too do not know what I am doing, but I'll be there. So if that means anything, we can come there and we can learn together what's going on. But it'll be a good time. I'm glad to be here. I work at the University of Georgia. I am in the Department of Entomology there. Jamie Ellis was my student. Okay, so there you go. Either thank me or blame me, but you know, there it is. That's our connection between our states. And but I don't think I've ever been down here, so this is the first. Maybe it won't be the last. I hope not. So what I want to do tonight is I'm going to be toggling back and forth between a couple different presentations because there's things that we're doing that are quite new. In fact, the date I'm going to be giving you a little bit today is the world premiere of some brand. I mean, you guys are getting it first, which is kind of cool. You know? So anyway, that I hope will be interesting to you. And I'm also going to cover the topic that you see up there in front of you, mutiny and other things that can go wrong besides mites. You know, my professional association is called the American Association of Professional Apiculturists, and we usually meet in conjunction with one of the national groups, either the Federation or the honey producers. We kind of go back and forth each year. And about 10 years ago, I realized, you know, we never talk about bees anymore. Do you ever get that feeling? You know, when you go to bee meetings, it's all these damn mites and bad queens and diseases and neonics and you know, everything but bees. And I started seeing this in my own professional group that you know, all the talks were about all of the pathogens and viruses and varroa, and I just, that's not why I got into it. You know? Is that why you got into it? No, yeah, really. So about 10 years ago, I kind of made a sea change in my own program at the University of Georgia and started going back and looking more at the bees themselves. What a novelty. And when we study them a little more in depth, we find that there's stuff there they can teach us that's really useful to know if we're interested in using our bees to maximize our honey crops or just ethically wanting to keep them healthy and happy. And with that, I'm going to jump to this cartoon. I know the print's a little fine, so I will read it for you, but this is a gem. A beekeeper sent me this cartoon a few years back, and I do not know who the person was. It was anonymous. If you are that person, identify yourself, because I have shamelessly used this ever since. But we have in the top left there a knight in shining armor. My queen, I pledge my allegiance. How can I serve the kingdom? Okay, here's what I want you to do. I want you to stab our enemies using this tiny sword. I also want you to hook the sword into your internal organs. That way, when you stab someone, it rips out all your guts and you die. Your enemy won't die, of course, but it should sting a little, and maybe they won't bother us afterward. Does this sound like a good plan to you? I love this. I mean, this is priceless, you know. Well, I hope at some point every one of you have had this kind of conversation go on in your head, because you know what they're talking about, right? This is the self-sacrificing worker. And why does the worker do this? It's one of the biggest mysteries in biology. And in another lecture that I don't have time to go into tonight, I go over this in some depth, that this mystery bothered the greatest of all minds in biology, Charles Darwin himself. And if you read in his great tome, The Origin of Species, he comes right out and he says, you know, this is a problem. This thing about the neuter worker, as he called it, this is a problem to my theory, because my theory of natural selection is all about a population like all of you in this room. You have variations in your genes, and some of them are good and some of them less good, and the good ones tend to accumulate just by natural extension of you living and able to reproduce. So over time, a habitat will shape the genes of a species and literally shape that species to fit very tightly into its habitat. This is Darwin's great idea. He put this to words for the first time. But he said the sterile neuter is a real problem for my theory, because they self-sacrifice. They don't reproduce. And to be a player in Darwin's game, you have to reproduce. That's one of the preconditions. And he went to his grave with this conundrum unsolved. And no doubt very frustrating. And the figuring all this out had to wait literally a century until a paper published in 1963 out of King's College in London kind of showed us how that can work. I'm going to talk about that briefly this evening, but just kind of keep that thought in your head. Why does an altruistic worker be altruistic? It's not just altruistic, it's the most extreme form of altruism. The worker is, here's what the worker is saying. The worker is saying, I am going to give up my own reproduction to help someone else reproduce. And if that's not altruistic enough, I will kill myself if necessary to do so. And that's pretty radical altruism. I want to perhaps disentangle some of the mystery behind that. And it is, of course, by the way, true. When they sting, it does rip out their guts and they die. And I remember when I was first getting started in beekeeping, I took some sweet sense of revenge in that knowledge. And when they pinned my shirt to my back, so to speak. Okay, with that, let's talk about, keep that thought in your head because I'm going to come back to that. Let's talk about things that can go wrong. And to begin talking about things that go wrong, let's talk first about things that go right. Now what you're seeing here is a schematic of what is understood to be the hierarchy of biological organization on Earth. And it starts from the bottom and works its way up. And there is a repeating theme in natural history on planet Earth that lower levels of organization lump together, and each of them subsumes their own genetic interests to the new group that they have now joined to. I'll show you an example. Right here at the bottom we have floating around in the primordial soup, so to speak, we have an unlinked nucleotide. Just the fundamental replicating molecule, if you remember from biology in high school or college. Well, these nucleotides don't get very far. If you're just a lone nucleotide floating out there, you're probably not going to last long. So what we saw was nucleotides lumping together into nucleic acids. And the most famous of them is, of course, that twisted ladder, the second one from the bottom, which we now know as DNA. And it really is like a ladder. Imagine a ladder, and then someone's holding it at the top and someone's holding it at the bottom, and we twist it. That's what the DNA molecule looks like. And it's simply a series of nucleic acids that form in conjunction next to each other, and they literally make up the alphabet of the genetic code. It's an information-based system. When I was going through college, as many of y'all, I can tell by the look of your hair, we're of the same generation on this. The hard sciences was physics and chemistry and mathematics, right? Because it was empirical, it was calculable, it was undeniable. The squishy sciences were kind of the humanities, right? And somehow biology got kind of lumped in with the squishy sciences. It was all just observational kind of stuff. That is no longer the case. Biology has unquestionably graduated up into the hard empirical sciences because of that molecule there. It is information-based. And this is where our genes reside on these chromosomes. And in each one of our cells, we have machinery in our cells that can read that genetic code and then create the exact protein that code is coding for. And it's manufactured in the cytoplasm of the cell. All of this action is going on inside the nucleus of the cell. Well, once again, a floating strand of DNA out there in the primordial soup isn't going to do very well. It's vulnerable. I mean, it can only contain so many genes for so many structures, and it's just going to fall apart. So what we saw instead was unlinked nucleic acids joined together into chromosomes bound in cells. And we have examples of this where it can be replicated in the laboratory, where these clumped nucleotides will spontaneously form protein cases that encase them. So once again, they've moved up into one level larger of organization. Well, a single cell, well, things are getting a little better now. We got that membrane around us. And in fact, it is such a successful form of life that it probably still dominates, well, it does. It dominates life on planet Earth. We call them bacteria. It's a very successful life strategy. But again, that clumping of lower levels into higher. Okay, well, where's this train going? We have simple cells that we see there in the very middle of the diagram that start lumping together. Because more cells looped together into multicellular organisms can now specialize and do more things. They aren't just a passively floating cell floating around in the ocean. They can now start getting specialization. Some cells can do different things than other cells. Some cells are, become specialized for taking in nutrients, detoxifying, mobility, the first flagella to float and move around. Multicellularity allows for specialization. And that's where we are today. You and I are multicellular animals. This is the level of biological organization that we occupy. We do not occupy the highest level of biological organization. Radical thought, that one. The honeybees do. Cool, huh? Because what happens next? You have a bunch of multicellular organisms who then lump together and subsume each of their selfish genetic interests to form a superorganism. And this is the highest level of biological organization that we have. Some people speculate, well, wait a minute, isn't it possible that we can have landscape scale organisms, superorganismal habitats, for example? And the theorists kind of come back and say, nah, we're not seeing that. So superorganism, at least at this point in our understanding, is the highest level of biological organization. In order for every step of that to happen that I described, there has to be a tacit agreement by all the members that, you know what, number one, we're going to get along. And number two, I'm going to subsume my selfish interests to the benefit of the larger group, and we're going to relegate all the reproduction to just one of us. And in your and my bodies, we have either ovaries or testes, which are the specialized cells for reproduction. And all the rest of our cells are in obedience to those germ cells, we call them. Our bodies, our minds, our limbs, our organs are vehicles to carry on the eternal genome that lives inside the germ cells of each of us, our testes or ovaries. And that's a successful body plan. And organismality, which is the noun form of the word, has been very successful. When Darwin was thinking about natural selection, it was organisms that he had in mind. The taxonomic nomenclature that we use to identify animals, Apis mellifera, Homo sapiens, it's all based on organisms. So it's what we have in mind when we think about how life is organized on Earth. And we still think about it with the honeybee. We think about it and we betray that bias with the words that we choose to use. Social insects. Here's another buzzword for you tonight. Social insects are falling out of fashion. They're officially falling out of fashion in the entomology world. Because think of it, these are terms that are medieval in their origin. Well, there are a bunch of insects, a bunch of individuals living together in this society. Look at them, there they are, living in one box. As I saw, ooh, there's the queen. It was very easy for medieval observers to just simply, translate their understanding of human societies and paste that onto the insects. Who's the big cheese in the colony? It's the queen. Originally, it was the king. But then they figured out that was wrong, so they made it the queen, grudgingly. But you see, this whole lexicon of words that we use to describe the social insects shows that we're thinking about them still as a society like us. But they're really not. The superorganism is the unit of selection. So getting back to Darwin's conundrum, the puzzlement that we each feel about a worker sacrificing herself for the good of the colony is no more alarming than when we slough off some cells. However many millions of them do we lose every day just walking around, There, I just lost some cells off my fingertips. Yeah, we lose them all the time. New ones generate, old ones slough off. And the loss of a worker is analogous to that. So this is just useful, and I have many, many reasons to think that it's useful for beekeepers to think of bees as superorganisms instead of a box of bees. Let me give you an example. I believe that one of the worst things of engineering that's ever been fobbed onto the beekeeping industry is the Boardman entrance feeder. And one of the reasons I think of that is because if you go back and look at AI Root invented that little gizmo, and if you look at the original catalogs with the Boardman entrance feeder inserted, they have a little pint jar right there. And I think that pint jar just impressed upon beekeepers that the units that we feed our bees syrup with are in pints and quarts, when it should be gallons. And if we think of that colony as not just a box of bugs, we're going to realize that that biomass is comparable to a medium-sized dog. And I would sure feed my dog more than pint increments. He looks at two. Kind of a big Gordo thing. But if we think of that as a biomass of a medium-sized dog, we will feed it better. Okay? We will understand the annual swarm cycle, because it's the superorganism wanting to reproduce. We will understand why the superorganism does it at the worst possible time for us, which is the peak of the nectar flow. It's the worst possible time. Don't swarm now, swarm, in September or something. Well, why do they do that? Because they're splitting their body. The superorganism is splitting itself in two, and this is a traumatic event for the swarm. It's traumatic for the parent, so they have to do it early in the season to give enough time for both of them to recover. It helps us understand things like overwintering, which I know is not a big deal down here, but it's kind of a big deal in Athens, and it's a real big deal up in Chicago, okay? But the bees do it, and they don't take the easy way out. I'll tell you who does take the easy way out. It's the bumblebees. Here's what the bumblebees do. At the end of summer, they hold a conference in their nest, and they say, winter's coming on, and what are we going to do about this? And they all decide, well, here's what we're going to do. I'm going to die. You're going to die. The males are going to die. The queen's going to die. The only ones who survive winter are the new virgin queens, okay? That's what we're going to do. And that's what they do. Okay, we all die except for the queens. And they hibernate. Well, hibernating is kind of taking the easy way out. They just die. Okay, we're all dead except for the virgin queens. They singly hibernate over winter, come out of spring, January, December, if you're down here, February up my way. They come out and they have to single-handedly forage for everything. They have to find a nest, they have to forage for food, they have to build the comb, they have to incubate the brood. They incubate their brood just like a bird does on its eggs. Just kind of hunker down over it and warm it up. That poor queen has to do everything. She slips back into a solitary, primitive stage, and only when her first clutch of workers emerge does she step her toe back into the eusocial stage. So this is what we call simple eusociality, where they kind of pass in and out according to the year into solitary or social living. Honeybees don't do that. They are superorganismal 12 months out of the year. So they have to have a very sophisticated way of surviving winter. They do it by clustering. I'll tell you another reason why superorganisms are an accurate way to think about the bees. It can lead scientists astray. About 100 years ago, there was a USDA bulletin published that was brilliant and popular, and we still see evidence of this bulletin in textbooks today. And what they did, they made these thermocouples. I think it was done up in Iowa. And they put thermocouples inside overwintering colonies. They had a thermocouple right in the dead center of the hive, one at the edge of the hive, and then of course they had an ambient thermocouple, and they just tracked it over the course of winter. And they found when the ambient temperature goes down, the temperature in the cluster goes up. And Obviously, this is some kind of a social exchange that's happening here. The bees at the perimeter are sensitive to this plunging ambient temperature, and they communicate that information to the bees in the core, who respond by revving their engines, which if you're a bee means you shiver your flight muscles without actually flapping your wings, and that raises the heat and dissipates through the nest and altruistically makes everybody warm, which is baloney. Absolute baloney. It does not work that way. I'll tell you how it does work, and it's one of those, duh moments. If this were a meat locker, this room, and I went over the wall and cranked it down to 0, what would we do? Yeah, exactly. We'd kind of, our interpersonal space would get like this much, So here we are, we got this human cluster in the middle of the room, right? They're clustering, right? Well, what's going to happen in the dead center is somebody's going to start saying, getting a little hot in here, make way, make way, make way, got to cool down here a little bit. Each one of us would find our own sweet spot in that cluster based on our age, based on our physiology, you name it. And that's exactly what the honeybees do. We don't need this highfalutin, fancy, complicated, what's the philosopher? Help me out here. The simplest explanation is always Occam's razor. Remember Occam's razor? The simplest, most economic explanation is the first one that we should probably always go to. We can't make that a universal pronouncement, but I think for the most part that's a pretty good one. And this is what happens with the superorganism. And you get this homeothermic temperature control that's just as fine-tuned as a homeotherm like you and me. So this is not the main point of my talk this evening, other than just establish in your minds that there is a biological hierarchy, and that it is repeatedly characterized with lower levels subsuming their selfish interests for the good of the greater whole. What's in it for them? What's in it for them? Well, usually partnering in natural history works. If I join forces with you, neither of us are really losers. We get to help each other propagate our own genomes. And I think that's an important point to make about evolution, because a lot of times there's a caricature of it that is in currency, that it's all about unbridled selfishness, when the flip side of the coin is equally true, that except for the viruses, evolution has found that partnering and cooperation has been a winning strategy. So that side of the coin needs to be said as well. For purposes of our meeting tonight, though, mutiny and things that can go wrong, let's refresh ourselves on some things here. These are chromosomes. Chromosomes are simply that DNA molecule that's been wound really, really tight. I probably shouldn't have passed that up so fast. See it there at the bottom left? See, if you were to grab the end of that chromosome and pull it out, you would see it's one big, long chain of DNA, that twisted little lattice where all of our genes reside. And their numbers, they invariably occur in pairs with what we call diploid animals like you and me. You and I, each of us inherit one chromosome from our mother and one from our father. And we call them homologs of one another. And in the formation of our eggs and sperm, those chromosomes will pair up, largely size to size. They clump together in roughly equal sizes, and they have diagnostic numbers according to species. You and I have 46. 23 from our mother, 23 from our father. Honeybees have 32. 16 from their mother and 16 from their father. But in the order Hymenoptera, of which the bees are a part, the ants and the wasps and the bees, the males are peculiar because they have only one set of chromosomes. They have exactly half of their mothers. They have 16. And this sets up some interesting relationships in the nest. If you have a sibling, a biological sibling, you share 50% of your genes with that individual. Both of you also share 50% of your genes with your common parents. Those are not the same 50% of genes, okay? Because when gametes form in our bodies, the sperm and the eggs, these chromosomes line up in a fashion like I'm showing you there, where the pairs come together, and they exchange genetic material. They cross their legs. It's kind of, sensual, if you will. And they exchange sections of chromosome and then pull apart again. And so these gametes, they then go on to separate and form their respective egg and sperm. But that's why the genes are always scrambled up. Have you ever wondered, how could I possibly be related to you? I know my sister told me that. Well, that's how. That's why. That's why we're not clones. We're siblings. We're not clones. You share 50% of your genes in common, but they're not the same 50%. It's kind of weird, I know, but that's how it works. It's a bid for a lot of genetic variation in each one of us, because it's another rule of nature that on average, nature rewards genetic variation. Now, this is a messy chart, but I'm showing you what the relationships look like in a honeybee colony in which the mother has mated with only one male, which is very rare and usually does not happen, but it was the norm in early evolution, and it still is the norm today with solitary species. Now, to make it more comprehensible, I'm going to do away with some of the peripherals, but take a look at some of the possibilities here. A mother up at the top, mated to one male, she will produce, a female daughter there in the second generation, and in the middle a male, and another daughter there on the right. And that female daughter at the far left in generation 2, she is faced with a choice. She's not a worker yet. Notice I've been calling her a daughter. This is early in the evolution from solitary to social living. She has a choice. I can reproduce. I can mate. I got a spermatheca, just like Mom. I'm still a solitary species. I could go out on my own and reproduce and do the whole schmeal of reproduction, or I could stay at the nest and help Mom produce siblings. Either way, I pass on 50% of my genes, because my siblings on average, my sisters and my brothers, if you average it out, I'm 50% related to them. Why not stick around? And that's exactly what they did. What we have in the case of the Hymenoptera, though, is this principle on steroids. Now, here's the math. Here's the only math I've got for you this evening, but it's worth thinking about because it makes some things click for you. In the Hymenoptera, where the males have only one set of chromosomes, what that sets up is a situation that's different than you and me. A daughter is not related to her sister by 50%. She's related to them by 75%. There we go. Okay, 75%. See that the bottom two daughters there connected together? Now, a female is related to her brother, which is her mother's son, by only 25%. But if you add them up together and divide by 2, you get an average of 50%. So at the end of the day, they're still passing on 50% of their genes, or sharing 50% of their genes with their siblings, just like you and I do. But the daughter can look at that and say, 50% if I stay at home and help Mom produce siblings, 50% if I go out and strike it out on my own. What would you do? You'd stick around in the nest, wouldn't you? This is where I always say I'm like a good millennial. My daughter is a millennial, so I can say that in good faith. Yeah, they're good millennials. They just stick around. Why? I'll just stick around and help Mom, because I can pass on just as many of my genes producing siblings as I can if I strike out on my own. And that is one of those crucial moments in social evolution. You remember Gary Larson, The Far Side? You remember that cartoon, Great Moments in Evolution? It was priceless. It's these two fish-like things, and they're right on the beach looking up they're in the water looking up at the beach at a baseball, and one of them's holding a bat. great moments in evolution. You know, you can just see them coming out of water to get back on land. Well, this is one of those great moments in social evolution. When daughters said, yeah, okay, I'll just stick around, Mom, is that all right? And then you get this situation happening right here. On the far left, we have scenario A, which is just a good old tried and true solitary bee. She's a lone ranger. She's passing on 50% of her genes with her offspring. She digs a hole in the dirt. She makes a little lateral tunnel off there to the side. She forages for a ball of pollen and lays an egg on it, and that's it. No parental care. It's over. Well, things have gotten a little different in scenario B, because at this point we now have a daughter sticking around. This is the very moment at which eusociality begins. This is the crisis moment. Will she stay or will she go? Well, she stays, and we have now lurched into eusociality. The daughter sacrifices her own reproduction and stays at home and helps Mom make more siblings. And we know this for sure when we get to nests like on C. Because if you look at C, we have 1, 2, 3, 4, 5 females, but only 2 brood cells, which tells us that not everyone is reproducing. And so it's between B and C we see that lurch into eusociality and the beginning of altruism in the bees. Well, so far that's fine, but this sets up all sorts of conflicts. If you are a worker, and if you are sharing 75% of your genes in common with your sisters, and you're only 25% related to you can't call them brothers because they're technically sons of your mother, because that's just, the males have no father. Remember that trickaroo? You know, males have grandfathers, but they don't have fathers. Okay, that's what's going on here. So if I'm a worker bee and I'm 75% related to sisters, 25% related to brothers, I am going to be in conflict with my mother because she wants to raise 50-50. She doesn't care. She's 50% related to her sons, just like she is to her sisters. Makes no difference to her. Every egg is 50% of her genes going on, and there is conflict in the honeybee nest. The workers and the queens disagree on how many females there should be in the nest. How do queens respond to this mutiny? They mate with multiple males. And why does that solve anything? Because when the mother mates with multiple males, yes, we're getting into the R-rated part of the talk here now. It's not even R-rated, is it? Yeah, if you start mating with multiple males, and if you start storing their sperm in the spermatheca, you now, and that's what you're looking at here, is actually these are worker ovaries, and right at the junction they do not have a spermatheca, whereas right at the junction of those ovaries in a queen you find a big fat spermatheca, which is an organ for storing sperm. Now the genetics inside that colony are different. I know this is messy, so I'm going to get rid of some of it too. Let's get rid of just some interesting stuff. Now, I am no longer related to all of my sisters by 75%. Some of them I'm just a half-sister with because we share different fathers. And these we call sub-sisters. And I'm only 25% related to them. Wait a minute, Mom. You snickered us over on this one. And it's not an accident that we do not see multiple mating in social bees until the two castes have diverged. Is that sneaky or what? And what I mean by that is you've got these females, one of which is the mother, and she has now evolved specialized queen-like characters. She can no longer do work. She's really good at laying eggs. The workers, on the other hand, or the daughters, were getting better and better at just doing work. And they could not reproduce anymore. The two were now codependent on each other. They could not survive on their own anymore. And it's only at this point that mothers started practicing polyandry. Sneaky, I don't know what the word would be. I'd have to invoke Spanish for that. But it's a sneaky thing that she's pulling off there, isn't it? because the workers would not have gone along with this. It's like, whoa, 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 whoa. I am no longer 75% related to my sisters. I'm only 25% related to a box full of subsisters. I'm out of this deal. Oh, wait, I can't, because I can't reproduce on my own anymore. To which the mom says, whoa. Conflict. It's a dirty, rotten trick, but it works. And it works because what's happening at this level is natural selection at the level of the colony. The colony is being rewarded for all of this because you now have a colony that is no longer genetically narrow. It's genetically radically wide. And this has made possible all of the behaviors that we enjoy so much about the honeybee, whether it's the dance recruitment language, whether it's their undertaking behavior, whether it's the sequence of tasks that they go through as they age, all the things that we read in National Geographic about, all is possible because of this great lurch in evolution, when the queens started practicing multiple mating. And there's conflict in sex ratio, and the only thing that makes it work is just sheer numeric tsunami. There's a whole lot more workers than there is that one mother, and they are making it possible to tip the sex ratio so strongly in the position of females. But it's a conflict. The queens would rather have it 50-50. It's workers that make the colony majority workers, sterile ones. Oh, it's exciting stuff. Who thought there was so much drama? Okay, so now I am a subjugated worker. I'm not happy with the situation. And what am I going to do? Well, you know, I can help mom produce brothers, and I'm related to them 25%. Okay. I really don't want my subsisters reproducing, because I'm only related to them 12.5%, which is even worse. So I'm going to eat your eggs, sis. And that's exactly what they do. What you're seeing here is a picture taken by Francis Ratnick's, University of Sussex in UK. He's the guy who's kind of done all of this. And they practice policing. The workers will try to cheat. Give them a chance, they will try to cheat. And you know exactly what I'm talking about. Anytime you've had a queenless colony, I mean, they jump on it. I mean, they are waiting for mutiny. The only thing that's constraining them is the pheromones of their queen. And so, if I can't reproduce, I'm just going to eat your eggs so you can't either. And they police one another. They go inside the cells. They can identify a worker-laid egg, which workers will try to do. They try to get away with it. But their sisters come in behind them and eat the egg. This happens all the time. It is the glue that holds the society together. Because anytime they stopped this mutual policing, all of the workers would go rogue and start reproducing and say, you know, forget this superorganism thing. Yeah, you can have it. I'm going to go out and I'm going to reproduce. I can't mate anymore, but I can sure activate my ovaries and lay eggs. And those males are perfectly fine, just as good as mom's males. So you got it. I'm going to show her. Okay? Workers are always primed to do this. And this gets us to another thing that can go wrong in the honeybee superorganism. And this goes back to my earliest theme about each of these stages must, by definition, relinquish selfish interests. You know, the nucleic acid has to stop being interested in only the genes that it carries and tolerate the genes that the other nucleic acids in that organism have, because we all kind of cooperate and get along. That's a tacit agreement at every step. But if those controls that are set up to keep that agreement in place fall away, then at every level we see those lower levels ready to rebel. In the honeybee, we call that laying workers. In the organism, we call it cancer. And it's the exact same phenomenon. It's always there, waiting to break the bounds and be selfish again. And this is another reason why studying the superorganism, which we can dissect and put back together again and not kill it, and other such benefits in biology. That's why studying the superorganism is so useful, because it sheds light on all the rest of the levels of biological organization up and down. And I think that's useful. I think it's nice to know that the things that we keep in our hives are the keepers of such secrets. But here we have it, cancer in the honeybee. You've seen it. Laying workers, they're rotten egg layers. They've kind of forgotten how, evolutionarily speaking, they're not good at it. They will stick their rear ends in, plaster an egg. They don't really know where to put it, but they do it anyway. And so you get these multiple eggs going on. And if you are a superorganism, well, I kind of misled you a little bit. There might be a level higher than the superorganism, but we think at this point that it's a maladaptive thing. It's a form of social parasitism. And we're seeing this in South Africa. With, if you've read the bee books, you'll know them. Apis mellifera capensis, the Cape bee of South Africa. This line here is showing that little red dot. The native range of Apis mellifera capensis is right around Cape Town. If you've ever been to Cape Town and you just kind of look around the bluffs around it, you're just pretty much looking at the full entire natural range of Apis cerana. It's a tiny little natural range. And then what you see, what began happening in the early 90s, beekeepers in northern South Africa who were keeping scutellata, which is our Africanized bee that we call it in this hemisphere, started moving their bees south in migratory fashion, just like we do in this country, and they got some Cape bees moving into their colonies. And surprise, surprise, these Cape bees are social parasites. The workers can produce fertile eggs. They cannot mate, they don't mate, and they don't have a spermatheca, but through what we call parthenogenesis, they produce a diploid egg, which produces a female, a perfectly functional, normal female. And these Cape bees are parasitic on other colonies. Trust me, we do not want these guys in our country. These are bad news. Because what do you do about it? You go out there and you see a lousy queenless colony, and they're all rotten. They're horrible, pathetic. But the difference with the Cape bees, they don't die. After a while, they will phase back into a queen-right phase. They will just take one of those worker-laid females, feed it royal jelly, she mates, and the engines ramp up again. Now the colony's good for a few weeks, and then it phases back into the queenless, and goes in and out, and it can infect other bees and parasitize them so that they go into this endless downward spiral, because those bees cannot resist the Cape workers, like some Cape bees can resist other Cape bees. Evolution is constantly morphing. It's the true arms race, shall we say. So, yes, in fact, there is a paper out there. It's talking about whether we are witnessing in real time a social parasitism with this Cape bee that is kind of dissolving the superorganismal structure and going back to a more simple solitary structure. It is not, based on our observations, it is not accruing into a higher level of organization. It's dissolutive. Evolution is impartial. It's not out there just to make a super bee. It's just what works. And in this case, a dissolutive, or an act that's dissolutive to sociality is working in the Cape. Why? Why? Why should it work? Well, there's really only one idea that I've ever read that put out an idea. And they said, around the Cape, it's really, really windy. And if you're a Cape bee queen on her mating flight, there's a real good chance that you're going to be lost in the wind. So therefore, this subspecies evolved the ability for workers to lay parthenogenic eggs and kind of bridge the gap until they could raise a queen again. I don't know. That's just somebody's idea. But I don't have any idea that's better. So this is things going wrong. This is not good for the colony's fitness, but yet it's right out there. Let's skip on this. Well, there's other things that can go wrong. The queen obviously has the brass ring. I mean, she's won the jackpot. She's the only female who has specialized ovaries to crank out 2,000 eggs a day, and she has the whole machinery of the colony to help her do it. Well, the the colony is quite happy to have her. The colony wouldn't mind having more than one queen. And this is in fact what the termites and the ants do. The termites and ants, if you can imagine, they live in kind of a spatial structure, you know, in kind of a two-dimensional structure in soil, and it's potentially infinite in its space. There's no boundary on the edge of a Formosan termite colony. You know, they can get as wide as they want to and can. And it's in the ants and termites that we, to this day, see the largest and most complex colonies, I think, in part because they are unbound by a nest, like the cavity nesting apis are. And so imagine, if you will, you're a member of an acre-large ant nest, and at some little kingdom on the edge of the empire, there may be a queenlet that rises up and says, you know what, I think I can pull this off. And she makes her little fiefdom off to the side of the empire. And she's still contributing workers to the common pool, and there's another fiefdom over there, and another fiefdom over there. And this is the norm in ants and termites. And that's why they kind of own planet Earth. You know, they are, this is a nice little anecdote for your next boring dinner party that you're at. If you are holding a scale of all the ants in Earth, add to this side all of the vertebrates on Earth, the ants outweigh all vertebrates on Earth. So they own this place, okay? This has been a successful strategy. The honeybees don't do that. What do? They got this big, luscious bed of royal jelly that they float in, and they raise all these cells. The colony is optimistic. Maybe this time. But no, it never happens, does it? The first one out, she gets out. I don't have a laser pointer, but you'll have to bear with me. See the cell at the bottom left? It opened normally because it's open from the tip. So this first queen comes out, and then she goes down that row and kills every one of her rival sisters. And you can tell that because they're opened on the sides. And queen cells that are opened from the side have been aborted by their own sister. And it really is the queen. The workers do not help with this. That new queen does all the dirty work herself, going down the row, killing her rivals. This is pure selfishness. This is absolute 100% unbridled selfishness, and evolution has not stopped it with apis. I think in part, like I said before, because of the confines of a nest, there really is not room for two competing queens. That's just my own idea. I've never tested or nothing, but they don't do that. And you as a beekeeper have a way to measure the cost. This is a cost. Remember, evolution is not perfection, it's optimization. You can do this if you do a double queen manipulation, which is a cool thing to do. When I was a kid growing up in Indiana, I did this until I got tired. I was making too much honey. And I was a strapping 18-year-old kid, and I was getting tired of climbing a stepladder to harvest my supers. It wasn't fun anymore, okay? But you make bodacious crops of honey this way. So here's what you do. On the left, we have a strong overwintered colony. It's probably going to swarm if you don't do something about it. With a double queen switcheroo, you basically make a split. You take some of the brood, put the new queen, and put it in a brood box at the very top with a couple supers in between, the old queen and the rest of the brood down at the bottom. And the only difference between this and a regular split is that double screen thing. You stick that double screen, which is literally a double screen. That's just a precaution so they can't sting each other through the screen, and then provided an entrance. And there's a little flight entrance in the middle of that double screen. And you let the two queens recover, lay eggs, and when you have sealed brood emerging, hopefully you have timed this to coincide with the start of your major nectar season. Then you rip out that double screen and let them co-mingle, and now you are having the rewards of a ginormously huge foraging force under one roof storing big crops. And from the year 1937, we know from USDA research that larger colonies make more honey per forager than smaller ones. So this is the principle that we're exploiting here. But as a beekeeper, you are witnessing with your own eyes the difference. This is what the species gives up year after year after year after year in productivity because of the queen's selfishness. So, evolution is not perfection. It's just optimization, what has worked up to this point. And they've been able to sort of get away with queens being selfish. I want to finish with one other thought to kind of leave you hanging on something. We're trying to get a grant to study this because I think it's kind of cool. This is a paper that's published in 2005, and it's from Germany, a prestigious lab there. And in this lab, they found evidence for what they are calling royal patrolines. And a patroline is simply that family of workers sired by one particular drone. And it's equal to the number of drones that the mother mated with, on average about 12 patrolines. And these investigators found that there was one patroline in the colonies that they were studying that had a huge proportion of the queen cells that were reared. It was like this patroline was saying, raise me, give me royal jelly, I'll turn into a good queen. And that's kind of cool. I mean, wow, that's interesting. These larvae are somehow signaling the nurses, to take care of me. It's kind of selfish, isn't it? It's kind of a step in the selfish direction, which made them a little nervous. But the flip side of that coin is at least more interesting. They found this same subfamily virtually absent among workers. They did not raise workers out of this subfamily. I guess they eat them. But if they needed a queen, that's the subfamily that they went to rear queens. So they're calling it now these rare royal patrolines. Well, what does this mean for beekeepers? Well, harken back to what I was saying about the Kate bee. Kate bees, once they start laying their own eggs, the workers, stop working for the colony. It's like they have just abandoned, and this is universal across the bees. I don't know of any difference, stingless bees in South America, or I think this is fairly universal among the bees. When workers begin reproducing themselves, they stop working for the colony. No more foraging, no more brood feeding, no more anything. That's why the colonies tank, because everybody stops working. And this appears to be a step back toward more primitive maternal states. When, I'm not going to work for the colony, I'm just going to lay eggs, because that's what reproductives get to do. So I'm just not going to do that work anymore. That's kind of what they're doing. It's a selfish, repressive step backwards, so to speak, evolutionarily, back toward a more solitary phase. And these authors believe that these rare subfamilies, which occur in mellifera and Italian bees and carniolan, the bees that we like, these rare families exist, are attempts at parasitization. These are workers that are trying to say, raise me, raise me. And we as beekeepers have an insight into this. Supersedure cells. Okay, what's a swarm cell? A swarm cell is when the colony wants to reproduce and they're getting ready to split. Both the parent and the split need a queen, so they raise new queens. It always coincides with swarming. And so we get these good queens in springtime under the swarming impulse. And so if you read all the classic beekeeping books, Roger Morse, Steve Tabor, they always say raise queens by simulating a swarming situation, or at least raise good queens in spring when they're in swarming season, because the resources are really good and those larvae get fed a lot of royal jelly and they turn into the best queens. Supersedures, on the other hand, can happen any time of the year, not necessarily swarm time, and the conditions may not be very good, and so you get a crappy queen. That is the traditional explanation of why supersedures are, not very good queens. Well, Moritz and his crew in Germany say, well, you know what? Think about it. in a swarm situation, who decides the next queen? Well, the mother herself has a significant amount of control in that case, because she will preferentially lay eggs in queen cups. And we know statistically that eggs laid in queen cups have a higher chance of being actually reared into queens. But the colony swarms, and the mother flies away, and oh gosh, we're a really big colony, we'd like to swarm again. Well, now who chooses the next queen? The workers do. The workers do. And there's all these little parasitic larvae that are shouting at them from their cells, feed me, feed me, feed me royal jelly, give me royal jelly, not the one next to me, feed me, feed me. And this is what we get in a supersedure situation. The workers calling the shots. The queen's dead, she's gone, you mashed her, or whatever. The workers find a larva, raise it. Who knows it wasn't a parasitic one? And if these parasites carry a dwindling worker genome for doing work, then their offspring will not be productive. And this is why we think those patrulines have no workers at all. Okay? They have no workers at all because they're not even good at raising their own workers. They're really crappy bees. So this raises the possibility that even in Mellifera, Ligustica, Carnica, that we have this sinister thread of parasitism that's trying to burst out through the bounds, and it's probably not good news for the honeybee, nor the beekeepers. I think this is evidence, I think this suggests a more nuanced reason of why we should prefer queens reared under a swarm season, not because the conditions are better, but because the queen herself deposited the egg in a queen cup, and therefore there's no reason for us to think that it's biased toward the parasites. Okay, let me wrap this up, and then I want to tell you a little bit about what we're doing at Georgia, and then I will be done talking for this evening. Just, evolution's not perfection, okay? It's just, and that's the comfort about evolution. It is a sanction of what has worked. It's not necessarily optimum. Workers are always poised for mutiny, just like the cells in our bodies are always poised for mutiny. This gives us a context for understanding why cancer is so insidious. It is woven into our genome. It is structurally a part of us. That is why cancer is so hard. It manifests in the social insects as laying workers and selfish queens. Queens and workers disagree strongly on how many females there should be in the nest. And the workers win simply by their numbers. Queens don't tolerate rivals. And this is a real cost to the colony, not a fictional cost, it's a real cost. And it raises the question, is the only option higher than the superorganism just parasitism? Just destroy the whole, structure that you inherited? I don't know. It makes one wonder. And then lastly, supersedure queens might be parasitic. And I think this gives us a good reason to try to avoid them, if at all we can. Let me jump to the one little thing I want to tell you about with our research, the world premiere, right, on what we're doing up at UGA, and then I'll have some time for questions. I have been working since 2012. I had a sabbatical in England, in York, which is the location of that country's national bee unit. In the UK, the whole FARA organization is, I don't know, I'm not quite sure what's happening to them, but for a long time they had regulatory and research lumped together in one unit, and they were stationed in York, which is up there in the northeast corner of England. If you're a James Herriot fan, it is Yorkshire, where James Herriot, the veterinarian, had all of his wonderful stories. And while I was there, we were interested in this idea that queens multiply mate, and the fact that multiply mated queens invariably head up colonies that are just better. I mean, everything we measure is, well, I shouldn't say that. most of everything we measure with colonies, if the queens are really polyandrous, mated with a lot of males, they're better. They forage more, they make larger honey crops, they reproduce more, yes, swarm more, because that's Darwinian fitness, being able to reproduce, and they resist parasites and diseases more. Our little contribution to this pool while I was there in York was to demonstrate that polyandrous queens have colonies that resist varroa mites. And think about this. This is in distinction to selection. I am not talking about selection here. Classical selection. A beekeeper goes out, you measure the mites. Oh gosh, this colony has half the mites as this one, so I'm going to breed from this one. Okay, generation after generation. That's selection. I am not talking about selection. In our work in England, we just simply found random drone colonies and gave them, see, what did we do? I think we did 15 drones. We inseminated with the 15 drones, or 30 drones, or 60 drones using instrumental insemination. At which point, I was at a meeting in London, one of their clubs, and there's this little man in the back of the room who stood up at this point, and he asked me a question. He said, "But won't she pop?" That's a great question. No. What we do, we collect the semen, and then we mix it, and then we give each queen, a uniform volume. But the volume still constitutes either 15, 30, or 60 drones. And we found a significant reduction in those colonies that were inseminated with either 30 or 60 drones, significantly fewer mites than those inseminated with just 15. So this is pretty cool stuff. I thought, without any selection, what if we could do away with selection? Wouldn't you like to be able to do away with, if you've tried to breed bees, you can sympathize with me. Yeah, here's our data. We also found significantly more brood per bee as the polyandry increased there on the top. The bottom graph is showing you the mites, and extremely low numbers of mites in those colonies inseminated with 30 or 60 drones. So I was pretty excited at this. In 2017, I got a grant with my colleague at University of Delaware, Debbie Delaney from Delaware, working with Keith Delaplane. It's made a lot of jokes around my lab. But we thought, well, if this is true, then what happens if we cross it with pre-selected semen? I mean, it's like, maybe it'll just synergize and make dynamite, right? So that's what we're doing right now. Well, here's our design. We have 2 levels of polyandry, either 9 or 54, and those numbers mean nothing. It was just that I had 9 drone colonies, VSH drone colonies that survived, and I wanted the top number to be a multiple of 9. That's the only reason for those numbers. They have no biological meaning otherwise. And we crossed these virgin queens with either drones that were VSH, Varroa Sensitive Hygiene, pre-selected for this character by Johnny Thompson from Mississippi, or just ordinary wild-type drones. So you see we have every possible combination of polyandry and pre-selected semen. And if you think about it, what's our possible results? Well, we could compare the row averages, and if we saw significant differences between VSH and wild-type, that's kind of useful. That'd be nice to know. It would tell us if VSH is in fact barking up the right tree, or if we could just do away with all of it and just, like he said, fill her up with semen until she pops. Or we could look at column averages and say, yeah, we just here we go. 54 drones is what we want. I mean, we get better mite performance regardless of any selection. But what if we see differences within the boxes? And this is where things could get exciting. What if we had a synergy? And our design was able to look at that. Oh, so let me show you our very latest results. What we're getting is 2 kinds of synergy. What you see, the green bars are the semen from the VSH drones, either 9 or 54 of them. The orange bars are the wild-type drones, 9 or 54 of them. And we have a weird synergy going on, and this is a significant synergy. The wild types that are polyandrous are just rocking on bee populations, and you wouldn't be surprised, brood production parallels this. And these are significant interactions. I've only looked at these data for like 10 days, and I haven't really thought that through yet, why there would be a difference in the behaviors between genetic selection and wild types, but So far, there seems to be that. Debbie's going to replicate her trial this year in Delaware. She's getting the colonies ready right now. But what we did find a significant effect of was a positive synergy with VSH and polyandry. This is a significant statistical test that you're looking at. Mites were significantly lowest in those queens that were inseminated with 54 VSH drones. So in other words, the polyandry synergized with VSH to optimize mite control. I think this is useful. I think this is important. What this is suggesting at this point, with only 50% of our data in, is that it's crying for an intentional integration of polyandry with classical bee breeding. And we can control this with instrumental insemination. Part of Debbie's and my grant is looking at simpler ways to do polyandry, because not every beekeeper has an instrumental insemination device, though I do think every club should have an instrumental insemination device. Just store that one away and think about it. For one thing, it's a lot of fun. And number two, you can learn it. It's like bicycle. You either learn it or don't, but you can learn it. It's not that intimidating. It's not that expensive. Stereoscopes, $225 on Amazon. You can get a good stereo microscope, which is all you need. The devices, Crispin Given, who's a technician at Purdue up in Indiana, he makes them. And he's probably got the best in the world that he just came out with this past year. So they're out there and they're available. His is like, I think it's around $1,500. So it's within the budget of a club. It's within the budget of a club. But you might also could do it through drone saturation. This is something we're going to be testing. We're also looking at mixing brood. I was giving a talk one year about this, and he was, I forget his name, but he was an inspector for the state of Arkansas, and at the end of the Q&A time, he raised his hand and said, "Can't you get what you're talking about by just mixing the brood?" I thought, damn. He's right, you can. Because you think about it. Think about it. If you have an apiary, and every queen has mated with her own 12 drones, from wherever, whatever. And if you take brood from all of them and kind of mix them all up such that they all have combs of brood from each other, and if they have on average 6 frames of brood, minus the edge frames, which kind of get wasted, that's 6 times 12, 72. That's 72 patrolines. Now granted, it's temporary, and I actually have a postdoc right now who's trying to model this. You mathematically model it. If you do this, that wave of polyandry last in the colony, it's kind of cool. We're probably going to get no, it's actually more like 45 or so, because it's their whole lifespan that has to play out. But it's a wave. It crests and then it goes out. And so we're trying to model this to get an idea of how long would that temporary proxy of polyandry last. So this is the kind of stuff that we're doing right now.
That's the goal, to do mite control by mixing brood? Is that correct?
That's the hypothesis. Right. The question is mite control with mixing brood. Now, there are lots of voices intaking their breath to shout out, but what about disease spread? Yeah, you're right, disease spread. There may be disease spread. So we haven't cracked that nut yet. We're just looking at this principle first. And so, we're going to look at it and see. And hopefully I'll be able to report those data sometime to you in the future. Yes?
Altogether, naturally, I feel like, from what I'm hearing here, they're trying to fix it themselves. We're coming at them with a different angle. But at the same time, the natural bees are the ones that are winning.
Well, they're winning when it comes to brood and bees. And VSH is not performing as well with brood and bees. But we're seeing the product of Johnny Thompson's selection playing out, and that benefit is synergized when we artificially elevate the polyandry. I think a question, too, to ask is, well, then why don't queens mate with 54 males? Well, this is, again, evolution. Well, they might pop, for one thing. You're actually right. Maybe they will. Maybe they will. But either they pop, or there's birds. You know, they're not very good flyers, so bird predation is always a risk. And sexually transmitted diseases, STDs. Deformed wing.
Is that a joke or that really?
That is for real. Yes, is that a joke? It's for real. Drone semen carries DWV virus, deformed wing virus. So, yes. Use condoms, he says.
The 12, 15, or 20 drones, and there's always like an excess amount of sperm that goes in there. It mixes, and then only a certain amount of it actually absorbs into the leak, and the rest of it's just discharged.
Yes, it's a huge amount of waste. It's a huge amount of waste. And you can imagine how the earliest polyandry was rewarded with those queens that probably, if you look at the spermatheca, it's a ball that occurs right at the juncture of the median oviducts coming, the lateral oviducts coming together to the median oviduct. Right at that, it's like a letter Y, and right at that juncture is where the spermatheca was. And I imagine there's queens that had like a little invagination there or something that caught more of the sperm and was rewarded with bigger populations and more genetically diverse little nests. And we can see how natural selection could have rewarded this on a gradualistic manner until it eventuated into the spermatheca that we have today, which is a really interesting organ. Actually, yeah, I'm listening.
Do you know what the average measurement is in microliters of the drone? What does the drone produce?
Yes, the drone is pretty much right at 1 microliter.
And how much does the spermatheca hold?
Well, it's not apples and apples because the spermatheca only has the sperm, not the seminal fluids. And that reminds me, oh, you gotta love the Brits room, so this is a good thing I'm saying about the Brits. But the Brits are really good at hobbies. You know, they are the hobbyist kind of people out there. They even have hobbies, clubs that take care of roundabouts. Roundabouts, for crying out loud. But the clubs that do that. Well, I was speaking just last October at the group in Devon, which is down in the south west coast of England, and I was talking about spermathecas and multiple mating, and oh yes, our club has collected data on spermathecas. I thought, what do you say? Yeah, well, we have a member in our club who's a veterinarian. He has access to microscopes, and there's a group of us that gets together every Saturday. Can you believe this? And dissects spermathecas. Try that for your next workshop. And yeah, so anyway, what I'm showing you here is not one of their pictures. It's one of my students, but I'm showing you that juncture. There, you can kind of see the letter Y there on its side, the two laterals, and then right there is what looks like a ball of yarn. That's the spermatheca, and all of that reticulation on the surface is tracheal tubes. It's heavily oxygenated to keep those sperm alive for the duration of the queen. And this is the picture that my friends in Devon gave me. They had sectioned spermathecas, and what you're seeing there is the outer, this is the membrane of the spermatheca that they have dissected, and it's only 1 cell in width. See those little black dots around? Each one of those is the nucleus of a cell, and it's only 1 cell thick. And all of that mass in the middle is sperm. And it is literally a ball of yarn. And the honeybee sperm is one of, if not the, I think it is the largest or longest tail of any sperm in the animal kingdom. It's just outrageous. It's stupid long. It's like this much tail and this itsy bitsy little head way up here. And how they get disentangled is anybody's guess. But that's what it looks like.
Shorter tails?
Yeah, maybe selecting for shorter tails. I don't know, but that's where the action is right there. And so,
You don't have to swim as far.
yeah, well, maybe so. Each little ripple takes them further, I guess. But what happens? I have people, when I'm speaking, they will say, "Well, okay, so does this mean Does this mean that I need to get queens from California one year and Florida one year and Georgia one year? And I say, no, you should get your queens from Georgia every year. But that's the wrong question, okay? That is not the variation that I'm talking about. That's true, I mean, Georgia, yeah, right. But their idea is right. If we have a variety of drones in our neighborhood, that's a good thing. That's never a bad thing. But I'm talking about the variation inside that spermatheca, which is a whole different can of worms. How many males did that queen mate with? And that's the hurdle that we're at, that at this moment we can only control with artificial insemination. By the way, drone congregation areas do a fantastic job at getting large numbers of drones. The best data we have are from the 1970s from Germany. I think it ought to be revisited because this is old classic mark and recapture, which is really fun to do, but nowadays we got genomic methods that would make it better. But they were tracking the numbers of males participating in drone congregation areas. You know what I'm talking about, drone congregation areas. These comets of males that fly in a circuit over square miles, over and over and over, looking for virgin queens. And this team in Germany found that the average drone congregation area had males representing 250 colonies. Wow. I mean, that's a boatload of them. They find them. The males find the route that all the guys are hanging out in. They find it. And so I think this is useful for another thing. We get twisted up, I think, a lot about our mating yards. Oh, well, where should I put my mating nukes and should I have my nukes 1 mile away from the drone colonies or should I have them be 2 miles? It doesn't matter. It doesn't matter. In fact, I would go so far to say that multiple mating, there's many things that contributed to multiple mating. I already talked about one of them, that it turns down the temperature on that sex ratio battle between the queen and her daughters. I would say a second one is also to compensate for brother matings. They probably do mate with some of their brothers, but when she's mated to 17 other males, it doesn't really matter. So there's lots of reasons to explain these selection vectors. Yes? Mm-hmm.
I think that's all that question really not solution, but I want to go for the whole point of when it comes to catching a wild swarm that you do box, that you hang on a tree or whatnot.
Yeah.
When actually done clean in there, would you suggest somebody as a newer beekeeper to keep that? And if she's doing well and doing everything proper, not aggressive.
Yeah. Well, that is always sort of the received wisdom, isn't it? There's two ways to look at that. She's a success. She's a self-evident success. Yeah. But at the same token, you don't know if that's maybe a secondary swarm, in which case it could be a parasite, based on what we talked about tonight. Yeah. And then, of course, you all have the whole, messy can of worms about the African bees, which we don't. So, yes.
As far as the queens flying out to go get mated, distance-wise, is a mile and a half, 2 miles?
Well, that's what I'm saying. I don't think it matters. Yeah. It is so outrageously outside of our control that it's a drop in the proverbial.
Is that the way it works? Do they really go out that far? Or?
That is based, I think that has been over in, yeah.
I think the question really is, how far does the queen fly to go get mated?
Yeah.
Well, classically they teach us that she always gets away from her apiary, but how far?
Yeah, the question is, how far does a queen go? And this is, I think, is one of those situations that's emergent and not intentional. And by that I mean, whoa, bad idea. Yeah, what I mean by that is she is not saying, okay, I am adapted to go a kilometer away. She's adapted to find a drone congregation area. and that drone came nearer or further from her. And I really am suspicious of those data. Those data, in fact, were collected in the context of Africanized bee work with USDA back in the 80s, when everything was about the killer bee, and they were interested in trying to measure relative flight distances of African bees and Europeans. And that piece of information has just been drilled into our brain, and I think it's grossly overstated. I'm kind of dubious about that. I think it's more driven by where are the drones? Ultimately, that's what matters. Where are the drones? Yes.
I have a hard time believing that with all that they are and that they can do, that a queen bee can't recognize her brother in a drone congregation. It just doesn't make sense to me that evolution would allow that to happen.
Yeah.
I don't know.
Yeah, I don't know if you all heard that. He's voicing skepticism on whether a queen cannot identify her brother and resist. I don't know. I think the question should always be posed. What can we measure? What can we see? And there's I honestly don't know of any studies. I know of studies that count patrolines. I don't know of any studies that have measured what you're talking about. Are one of those patrolines my brother?